So we talk about the two simple mechanisms of gene duplication. And in Long's et al.'s review, they put a big table of the gene duplication, the mechanisms of gene duplication. So I want to go through each one just to explain to you the idea, but they are all very similar. The first one is exon shuffling. So the idea of exons is because we look at our genome, in most eukaryotic genome, our genes are in pieces. They are not continuous as in bacteria. And we have introns. But the exon module itself, we can think about or we can hypothesize in the early days of evolution, actually these exons, each individual exons could be one of the genes. And now they are combined or assembled together to be specific in one function. Then the exons can be inserted into the genome. So here is the idea showing in a greenish blue, and that block get insert into the intron of a gene. And Manuel has a very remarkable paper study of the genome-wide, how likely this be the mechanism in eukaryotic genome. So if any of you are interested, you should look up that paper. So by exon shuffling, it means we can look at our proteins. Now where proteins have different functional domain. A lot of proteins have DNA binding domain, so that particular domain is a functional units. By exon shuffling, you can think about the protein domains can reassemble to form a new gene materials. So this is the idea about exon shuffling. And the second one is the gene duplication, and this is like the basic model. And the third one is the retrieval transposition, we just explained, because the messenger RNA get reverse transcribed and get into the other side of the genome. The fourth one is the mobile element, and this is also a recent discovery. We know we have transposable element. We know some transposable element has like a high copy number in certain species. For humans, we have allele, and allele has like multiple copies in human genome, and we can study the allele element evolution in a single human genome. But we also found sometimes the transposable element randomly inserted genome be recruited, be part of the gene content. So showing in this diagram is the idea. This blue, now the shape is a little bit different, but now indicate it's a transposable element. So transposable randomly insert into the genome, and this is the case. You can see it insert into in between the two exons and be recruited, be part of the gene. And so the transposable element at the end be transcribed and translated, become part of the protein. And the other one is what we haven't really talked about is later transfer. And later transfer, I think most of people would think is more or more often occur in bacteria, but also in higher organisms. So the later transfer means the genetic material was obtained from other species, not by their common ancestor, but by other mechanisms. So this is the later gene transfer. And gene fission or fusion, it means a gene could fuse together, two genes become one gene, or one gene separate into two genes. So this is like a mechanism, and the illustration is on the other side. And the other one, which we think is not possible, but we do have a few cases indicate the gene can be de novo generated. When we say de novo generated, because it can use a piece of intron or internogenic region and they become a transcribed or functional gene copy. So all this was summarized in the review paper, talk about possible ways to generate new gene function.